In this video, we're going to talk about the biology of Leishmania species. In the previous video in our parasitology, we talk about the Trypanosoma cruzi, which is American trypanosome. And we say that they are mainly found in America and not found in Africa. It is Trypanosoma brucei that we can find in what? That we can find in Africa. Now, remember that we divide flagellates into intestinal and uroflagellates and hemoflagellates, which means we have one is intestinal flagellates, we have other to be hemoflagellates. Now, hemoflagellates are blood and tissue flagellates. Let me just classify that one. So I said we have flagellates. Let me use that one to denote F, and they are divided into two. That is intestinal flagellates or urogenital flagellates. And we have what? We have hemoflagellates, right? Hemoflagellates. And under these hemoflagellates, they are the ones that live in what? That live in blood and tissue of what? Of mammals, you understand? Now, under these hemoflagellates, we now have two. They are mainly two. We have trypanosome and we have leishmania. Under trypanosome, we now have anterior station trypanosome as well as posterior station trypanosome, you understand? And we have talked about that one in our previous classes. Now, today, we'll be talking about the Leishmania species, which is also an hemoflagellate. It is similar to what? It is similar to trypanosome, but there are some distinguishing features that differentiate it from what? From trypanosome. So today, we're dealing with what? We're dealing with Leishmania species. Now, let us proceed. Leishmania species are flagellated protozoans that inhabit the reticulum epithelium, system, tissue, and cells. They inhabit the what? The reticulum, the epithelium, system, the tissue, as well as the cells of what? Of mammals. You understand? Now, the macrophages of lizard and man, and they causes the disease known as what? Known as leishmaniasis. So this is the disease that they cause. Hence, we can say they are what? They are pathogenic parasites because they cause a particular disease known as what? Known as leishmaniasis. You understand? So this is the disease that they cause. Now, leishmaniasis is one of the most disfiguring disease of man. Please just hold this word. One of the most disfiguring disease of man. You understand? Because they created something we know as saw. I'm going to show you an image which shows that. Now, the Leishmania species cannot be easily distinguished from one another morphologically because they are similar to one another. They are species. They are similar to what? To one another. But the distinguishing factor is based on isoenzymes, as well as this isoenzyme also solve the problem of taxonomic as well as identification of associated Leishmania species. You understand? Now, like I said, they cannot be easily distinguished. You understand? They cannot be easily distinguished because they look alike. But because of some chemical analysis, we can distinguish this one from this one. You understand? Now, we now have 7 complexes. Just the 7 basic complexes that we're going to talk about. They are more than that. They are very, very much. But the ones that we'll be talking about is Leishmania donovani, L major, L brasiliensis, L ethiopica, L genapsiasis, L tropica, as well as L infantum. These are the 7 complexes that we'll be talking about. You understand? Now, I said basically Leishmania species can be classified into 2 groups, just like trypanosome as well. Trypanosome, just like they are classified into posterior and anterior. We can also classify this one to Leishmania proper and Leishmania viana. Leishmania proper are those ones that develop in the midguts, and Leishmania viana are those ones that develop in the midgut as well as the what? As the hindgut. So example of Leishmania proper is L major and L donovani. An example of Leishmania viana is L brasiliensis. You understand? Now, let us now proceed to the structure of this animal. They are spindle shaped just like trypanosome. Look at it, they are spindle shaped. And they have what? One thing about them is that look at their kinetosome. Can you see that their kinetosome is what? Is anterior. And their nucleus is what? Is central without undulating membrane. Can you see? They have no undulating membrane. And this is their structure. This is flagella. This is kinetosome, and this is their central nucleus. Same thing with this one. You understand? These are the structure of what? Of Leishmania under the microscope. So these are the basic structure of Leishmania. But one thing we just have to notice that they are also spindle shaped. They possess kinetosome, they possess flagellum, and they have a central nucleus. You understand? Now, talking about their etiology. Etiology of Leishmania species can be caused by many species of Leishmania, which is a protozoan parasite in the subfamily of Leishmaniae, you understand? Now, and a family of Trypanosomatidae, order of Trypanosomatida. They are still under Trypanosomatidae. You understand? Now, we now have different this thing. We have, like I said, we have Leishmania viana and we have Leishmania proper. And under this Leishmania, we have L major, which is Leishmania major, Leishmania tropical, Leishmania atropical, Leishmania donovani, Leishmania infantum, Leishmania mexicana, Leishmania amazoniensis, Leishmania venezuelensis, and others like that. So these are the parasites. These are their complexes that can cause what? That can cause the disease known as what? Known as Leishmaniasis. You understand? These are their species, their complexes that can cause the disease known as Leishmaniasis. Now, this is a table that shows the what? That shows the classification of Leishmania in both Old World and the New World. You understand? So basically, the genus Leishmania can be classified into. Remember that we have their complexes. Can be classified into Leishmania, Viana, and Cyro-Leishmania. But the ones that we are interested in this class is Leishmania and Viana Leishmania. You understand? These are the two that we are interested in this one. So under the Leishmania, we have the Old World cutaneous, we have the New World cutaneous, and we have the visceral Leishmaniasis. But don't worry, we're still going to talk about the pathology of Leishmania species. Pathology of Leishmaniasis. They cause the disease. They cause cutaneous Leishmaniasis, mucocutaneous Leishmaniasis, and visceral Leishmaniasis. So we are going to talk about that. You understand? Now, let us move to their life cycle. The life cycle is indirect, or that is, they are heterogeneous, which involves two hosts. The first host is the sandflies, and the second host is the mammalian host. I will show you the image of a sandfly. Now, the life cycle begins when the vector takes the blood. You understand? It takes the blood of an infected mammalian host. You understand? Now, what it takes in is a flagellated A mastigote. That is what it takes in when it is blood feeding. When it is feeding on blood of their host. You understand? Now, the A mastigote, immediately that it is taken in, it migrates to the midgut, where it transforms into a weakly motile promastigote. That is the case of L proper. That's the case of what? L proper. Remember I told that we have Leishmania proper, and those one develop in the what? In the midgut. So it transforms into the what? Into the midgut. But what about L viana? L viana, they will migrate into the pylorus or the ileum of the hindgut, and in that place they will what? They will multiply. Remember I told that L proper is midgut, L viana is from midgut to the hindgut. You understand? So they transfer into the what? Into the hindgut. Now, the weakly motile promastigote, that is this one that is here. So we are continuing like that. The weakly motile promastigote subsequently transform into a rapidly motile promastigote. Now this promastigote is now called nectomonad. You understand? It is called nectomonad promastigote. Now nectomonad promastigote now migrates to the foregut. You understand? And it develop into a replicative promastigote that is found attaching to a stomodial valve of the what? Of the food channel. Are you getting me now? It is found attaching to what? To the stomodial valve of the food channel. Now this type of promastigote produces a gel, and we know them as promastigote secreting gel. Are you getting me now? Promastigote secreting what? Secreting gel. Now we now proceed. This promastigote secreting gel now develop into what we know as eptomonad promastigote, which later divide and transform into a metacyclic promastigote. Now this promastigote is the infective form to the mammalian host. Once the sandfly comes again to feed on the blood of another man, to feed on the blood of another mammalian host, it now injects this metacyclic promastigote into the blood of the man. Are you getting me now? Now this metacyclic promastigote now undergo binary fission, which means it divides. You understand? And when it divides, it becomes a non-motile amastigote, which in turn multiply by what? By binary fission. And it is this a mastigote that the another insect or another sandflies will come and take during blood meal, you understand, and that is how their life cycle proceed. Now this is an image that shows the life cycle. Now look at it. The sandfly takes look at it. The sandfly, let me start from here. Now look at it. This is promastigotes are phagocytosed by what? By macrophages. Now, the promastigote now transform into what? Into a mastigote. So once they transform into a mastigote inside the cell becomes filled with a mastigote. What happens? You know, this is a cell. It becomes filled with what? Let me use that one to represent black. It becomes filled with a mastigote. Look at it. There is a mastigote inside. So it becomes filled. Then it ruptures and what? And bursts to release what? To release a mastigote into various tissue to infect other cell. You understand? So it is now this a mastigote that this sandfly will now take in. You understand? It will take it in. Now, once it is taken in, it will transform into promastigote. Now, remember I told you that we have different promastigote. We have nectomonad, we have leptomonad, we have ectomonad. You understand? Now, this a mastigote now divide. You understand? They divide in the gut, either the mid gut or the hind gut. When they divide, they develop into promastigotes. You know, I told you that promastigotes. So they develop into metacyclic promastigotes. So it is this metacyclic promastigote that now migrates into the proboscis. That is the foregut. Remember I told you that they migrate into the foregut. Now they now go into the proboscis of the what? Of the insect. So the proboscis, when the insect is now feeding on the blood of a man, you understand, it's now what? It deposits the metacyclic promastigotes into the what? Into the blood of a what? Of a man, and that is how their life cycle continues, you understand. Now let us proceed to the epidemiology. The disease is common in low income community. So we can call it the disease of the poor. You understand? Disease of the what? Disease of the poor. That is low income community with primitive thatched house, thatched house, low standard hygiene, where organic waste, stone and bricks are being found. You understand? They are being found. Now, woman settlement that is close to dark holes. I want to write hole here, sorry. Dark holes, rodent bore, termite hill, cave, cow sheds. They can serve as resting site for the parasite vector, which means the parasite vector can be hiding in this what? In this site. Are you getting me now? So once the parasite vector is hiding in this site, it means they can easily transmit the what? They can easily transmit the infection. You understand? In a tropical weather, people that lives there, they tend to wear a light clothes, which expose them to the what? Which expose them to the vector's parasites. You understand? This is another one. You understand? Now, the vector of leishmaniasis can be phlebotomus in old world and Leishmania in new world, and their biology as well as ecology varies from region to region, but they have direct influence on the epidemiology of the disease. You understand? It is where they are much, that is where the disease is going to be much. Are you getting me now? They have what? They have direct influence on the epidemiology of the disease. Basically, sandflies, they feed on human blood, and they can feed on human blood in the dusk, in the dawn, and at night. You understand? Now, most leishmaniasis is a zoonotic infection. That means it can be transmitted from wide range of animals. From wide range of animals, they can serve different animals can serve as reservoir host. Example is dogs, rodents. Sorry, dogs, rodents, dogs, rodents, sloths, and so on. These are animals that can serve as what? That can serve as a reservoir host. I get me now. Now, other risk factors of leishmaniasis is the habit of rural peasants keeping poultry birds, dogs, sloths in a place that is not hygienic. You understand? In a place that is not hygienic. this can increase the exposure to sandflies. You understand? Emigration from non-immune individual, emigration of non-immune individual to a place that the disease is much. You understand? To a place that disease is much, and the habit of sleeping in sight of terrace without bed nets or any this thing, it can lead to what? It can lead to a risk factor to this particular disease. You understand? Now, moving from there, this is an image of what? Of sandflies. This is an image of sandfly taking what? Taking a blood meal from a what? From a man. Can you see? Now, this is the proboscis. Let me clean this. This is the proboscis. Can you see? This is the proboscis that is. This is the proboscis. Can you see? It is being injected into the skin of that man. So if it is going to take in, it is going to take in. If it's going to deposit, it's going to deposit. You understand? And this is the reason why I say it is one of the most difficult diseases of man. Look at it. It creates a sore. See, if it is mouth, that's how it's going to create it. If it is leg, if it is anywhere, you understand? But this one, as I'm seeing it, it is cutaneous leishmaniasis. It is cutaneous leishmaniasis because it is on this thing. So it is cutaneous leishmaniasis. So basically, in the pathological manifestation of leishmaniasis is depending on pathology of the species as well as the geographical zone and the host immune response. So it is depending on two things: geographical zone, geographical zone, and the what? Host immune response. You understand? So we now have three. We have three this thing that can be caused by it. We have the visceral lesion, visceral lesion, the visceral lesion. That is, they call it Kala-Aza. Kala-Aza. We have, this is number one. You understand? The second one, we have the cutaneous leishmaniasis. Cutaneous leishmaniasis. You understand? And they call that one oriental sore. And the third one, it is mucocutaneous leishmaniasis. They call it mucocutaneous leishmaniasis. Cutaneous leishmaniasis. You understand? This is mucocutaneous leishmaniasis. So these are the three diseases that is caused. So in the next video, we'll be talking about the pathology of Leishmania species. Thank you for listening, and subscribe for more. Thank you so much.